Marine Mammal Institute here and works with Bruce. And he comes originally from Colombia, where he did all sorts of great work working with the dolphins in the Amazon and birds in the Caribbean while he was doing his bachelor's degree. He then worked on his PhD here in Oregon with Bruce Mate, working on marine mammal ecology around the Galapagos, so all sorts of wonderful places. After that, he was a postdoc scholar with NRC, doing work on the California current, and he's here today to talk a little bit about work that he's done since he's come back to OSU on blue whales. So welcome and thank you.
Thank you, Cinnamon. Hi, everyone. All right, so hopefully all you hear is good. So the reason for this presentation today is to provide some of the results that we have from a grant we recently completed for NASA that essentially was intended to mine a number of the whale tracking data sets that we have here at the MMI. So at the very bottom there you see the name of all the collaborators in this grant, including Bruce Mate and Lad Irvine. So what you see here is the largest tracking data set of any whale species anywhere in the world. There are 104 blue whale tracks represented in that map. And it really is a testament to the vision and the dedication of Bruce Mate, who has been a pioneer in whale tagging for over 30 years and who started tracking these animals in 1993. The animals were tagged in 5 areas that you see there on the screen, hopefully circles with a darker circle inside, primarily in Santa Barbara Channel in Southern California, and in the Gulf of the Farallones in Central California. A few more whale tags were tagged of Cape Mendocino in Northern California, and in so those are for the summer part of the range. And then in the Gulf of California and the Costa Rica Dome during the winter part of the range. So collectively, these tracks have shown us the complete migratory range of the eastern North Pacific blue whale population from the summer and fall range along the west coast of the United States to the winter-spring range in the Costa Rica Dome, and also in the Gulf of California, as well as along the west coast of the Baja California Peninsula. So satellite tracking through the Argos system can provide 1 or more locations per day, but due to the coverage of the satellites at any given time and location, we can have different qualities of the locations. And that's what you see in the black circles and lines on that map there, where you see sort of stray circles. And these days we have statistical techniques known as state space models that essentially allow us to improve on these raw tracks by estimating locations that incorporate the error associated with them and improving them. They also allow us to produce regular tracks. So in this case, we in the red circles, we see a regular track with 1 location per day with improved location quality or location uncertainty around it. So we also know from animal movement models that an animal may display residence in an area where it may spend considerable time by performing frequent changes in speed and direction. So that's what you see in the bottom panel, sort of in that area where there's regular changes in speed, in direction anyway. And then the animal will move rapidly and in a linear fashion to a new patch and repeat that process. So we can infer from these area-restricted search areas at the 2 ends of that particular trajectory and the taxis space in between that they might correspond to transiting and foraging modes, which we see in the blue and red circles at the top. So that's another feature of the state space models, is that at any location they allow us to estimate a behavioral mode, either or. However, one thing to keep in mind is that these known as switching behavior state-space models have not really been validated in terms of what those behavioral modes really mean, other than the general theory about animal movement, that they generally tend to spend some time in an area and then move on. But that could apply as easily as to housefly in your kitchen moving from one food scrap to the other, or to a blue whale moving throughout the eastern North Pacific. So there's not a direct underlying implicit ecological process other than the movement model, underlying movement model in it. So one of the first things we did with these state-space model tracks was to analyze the continuous parts of the tracks that were identified as ARS behavior, and we call them ARS patches. And in one of the first papers from this project, we determined that the typical patch length scale was 50 kilometers, and the mean residence within a patch was 21 days. The mean transit time between patches was 14 days, and the mean distance between patches was 888 kilometers. And also, the mean distance between consecutive locations was 40 kilometers. So this information is key because it is what guides the kinds of questions that we can ask about these data and the scope of inferences that we can make with them. So once with that information, we also wanted to have a better understanding of how similar or different the animals were in terms of their use of the habitat. So Larry implemented a home range analysis, and what we see here is the overlap of home ranges. So he took the entire home ranges, or I'm sorry, the entire track for each animal within the U.S. exclusive economic zone, which is what you see in the black polygon encircling that area on the map on your left. And after computing the home range for each animal, what you see in the shading in the red to orange, yellow to darker orange shading is actually the overlap, how many of the home ranges overlap together. And we see 3 hotspots along the coast from north to south. The biggest one is, and the strongest one is of Point Conception and the Channel Islands in Southern California. Then we have one in the Gulf of the Farallones in Central California that is essentially outside of the San Francisco Bay Area. And then the third one is a more diffuse one and maybe more extensive one ranging from Cape Mendocino in Northern California to Cape Blanco in Southern Oregon. So the identification of these hotspots had important management implications because 2 of these occurred in the path of some of the busiest shipping lanes on the planet. And ship strike mortality has been a rising concern. So the spatial information, this type of spatial information is key for managers who are looking to mitigate this problem. So we now have an idea of where some of the main hotspots occur, and we wanted to know why they occur where they do. And that's the topic of today's presentation. So for this purpose, we took a subset of the data ranging in years from 1999 to 2008, which is the period when we have a full complement of environmental conditions to match to the tracking data. And like the previous study, also extracted locations just occurring within the exclusive economic zone of the US. And for the months that we know the whales are here feeding, which is July to November. So this gave us a subset of 64 tracks and 3,334 locations, and each of them had an associated behavioral state. You will notice, so that's the first map you see on the left, and you'll notice that that scale ranges from 1 to 2. This is because the state space model runs about 20 to 30,000 Monte Carlo Markov chain simulations, and what you see for each location is the average value of the behavioral mode estimated from those 20,000 runs. So that's why, even though we started with specifying either mode 1 or mode 2 corresponding to transiting or ARS, we end up with a continuous scale between 1 and 2. The next step was then to grid these locations using grid cell size of a 1/4 degree resolution. And this gave us 538 grid cells in that particular area. So that covered about 39% of the US EEZ. That's what the combined tracking data covers for that particular area. So then the next step then is to take that the behavioral state that we saw ranges between 1 and 2 from each location inside those grid cells and compute an average for each track, for each animal inside the grid cell, and then average that across all animals. And once we had that, then we can use the cut of 1.5 and say anything below 1.5 is transiting, and everything above 1.5 is ARS, which is the traditional way that these results are interpreted. So that's what you see here in red and blue, where red is ARS and blue is transiting. And the kind of question that we would ask from something like this is, given that a cell is occupied, how likely is it to be used for ARS behavior? So now we have moved from individual locations to grid cells. But, one more thing here. So even in this sort of gridded aggregated data set, we see that the primary hotspots that were identified from LAD's home range analysis appear here as ARS areas, which are the circles that you see here in yellow. Again, Point Conception area, western end of the Santa Barbara Channel, of the Gulf of the Farallones of San Francisco, and then the northern one from Cape Mendocino to Cape Blanco in Oregon. There is also an offshore area sort of west of Point Conception in Central California that we will get into a little bit more a little later. But let's step back for a moment and look at the physical and biological mechanisms that drive blue whale occurrence in the California current ecosystem. So we know that blue whales rely on krill as their only food source and require dense aggregations in order to feed their massive bodies. We also know that krill has a tendency to associate with certain bathymetric features that allow them to maintain position relative to the prevailing current flows. And this takes place especially at the continental shelf break, which is usually represented by the 200-meter isobath, and also around submarine canyon edges where krill find an ideal place to establish dense concentrations. So what you see in the figures here are schematic representations of how krill patches during the day on the map, on the figure on the left, that is what you see on the x is distance along a transect from moving offshore to inshore, and then in depth on the y-axis. And what you see is a patch of krill right at the shelf break around just between 150 and 200 meters depth. During the day is in areas of low chlorophyll or low phytoplankton concentration, but then at night they would migrate up to the surface where they will feed on the higher phytoplankton. And then because of the current flows, we have two types of current flows: a cell that's essentially circulating offshore to inshore, and then another cell that's circulating northward and southward depending on depth. This strategy allows them to generally maintain position at a given place to optimize their foraging. Another concept that's important to take into consideration here is the wind to whales idea that was advanced by Don Kroll to explain the seasonal appearance of blue whales in the Monterey Bay coastal upwelling ecosystem following upwelling events, large phytoplankton blooms, and then krill. So our objective today is to explore how bathymetric and oceanographic conditions combine to produce favorable habitat for blue whale foraging. So we took 5 bathymetric predictors derived from digital elevation models. Those are depth, slope, eastness and northness, which is the decomposition of the aspect of the slope, into two separate variables, and then the distance to the shelf break. So that's what you see in those top 5 maps. And then at the bottom 4 maps are oceanographic predictors, including sea surface height, Ekman upwelling, sea surface temperature, and chlorophyll. It is important to note here that these variables have been detrended by latitude. The reason why is that a lot of these physical and biological oceanographic variables have a very strong latitudinal underlying pattern to them. And if you don't remove that, what your habitat model or any correlation that you try to establish will be primarily capturing the latitudinal gradient, which is very strong, rather than the real oceanographic pattern or process. Okay, so we wanted to examine these relationships formally, so we used the habitat model. And in this case, in particular, we're going to model behavioral mode as a function of environment, where the response is a binary presence or absence of ARS behavior, and the predictors are environmental proxies of krill aggregation. In this particular iteration, we use non-parametric multiplicative regression in hyper-niche. And what we have here are the functional responses that represent the best model identified by 4 predictors, including the eastness of the slope. So that is essentially how westward or eastward any given slope of the seafloor is facing, the distance to the shelf break, the detrended sea surface temperature, and the degree of the slope. So we see that ARS likelihood was highest in the most westward facing slopes that were close in proximity to the shelf break. The sea surface temperature response is actually bimodal with a peak at negative SST anomaly, which indicates the coastal upwelling centers that we're familiar with right along the coast, and also areas with slightly positive anomalies around maybe like 1.5 degrees above the surrounding waters. That they actually correspond to offshore waters that are found downstream of upwelling centers. So these waters have had some time to increase their temperature, but they are still associated with that sort of rich upwelling environment. And then finally, in terms of the slope, we see that the likelihood of ARS was highest in intermediate high to intermediate low slopes, but not in the lowest or in the highest of the slopes. The primary measure of fit for a binary response in non-parametric multiplicative regression is what you see there as log B, which is a value of 8.2. Relating to a specific value is not easy without a larger frame of reference. But I will just say that because it varies by sample size. So we had a sample size of about 490 grid cells that entered the model. And in this case, it's relatively low in terms. You can see it's an analogy to the deviance that you would get from a logistic regression type approach. Another way to think of this is the average B metric of 1.04. What that is telling us is that an average grid cell is contributing about 4% to a model that is better than essentially a random model. The tolerance and sensitivity that you see there, the tolerance essentially is ranking the importance of the variables. So eastness is more important than distance to the shelf break and to sea surface temperature and to slope. And the sensitivity is telling us how much you need to vary one of those variables for it to have an impact on likelihood of ARS. So again, eastness is very sensitive, whereas sea surface temperature can vary the most without that having an impact on likelihood of ARS likelihood. So these are the projections, the full predictions of the ARS likelihood, and this actually looks pretty good. we see areas that are predicted where we know that there's increased likelihood of ARS. And those were, again, what were identified before, the known hotspots from LAD's home range analysis, and also that offshore area that represents sort of the extension of the California current offshore, where there's still high productivity and probably large euphausid or krill abundance. And then what you see in the panel on the right is the same representation, the same predictions, but it's a density distribution to show how the model doesn't really predict likelihood above 80%. So that's essentially like thinking about a prediction of how we would expect a weather forecast for tomorrow. You know, is it, do we know that it's going to rain with 100% certainty or 50%? In tomorrow's case, I think it's going to be about 30%, I can tell you that, here in Newport. But what is good here is that you see how it peaks above 0.5. And although this is not necessarily encouraged, a very common practice these days with binary type regression or any type of binary habitat model is to classify the response or the prediction, the prediction of the response rather, into either presence or absence, just like we had the input data. And we're going to do that exercise here, although it's not necessary, but to do that exercise, we can take the 0.5 or 50% threshold as a cutoff to say anything above 0.5 can be classified as presence, and anything below 0.5 can be classified as an absence. So now this is what we get. And what you see there in the table is what's called a confusion matrix, where in the rows, or actually on the column on the left, what you have is the true observations, and the other columns are the prediction. So that is essentially telling us how the prediction and the observations are being represented. So the dark blue are what's called the true negatives, where both in the prediction and the original sample they were absent. And the dark red is the true positives, where both the prediction and the original data had presence of ARS behavior. And then the light blue and the light red represent the false positives and the false negative. And that's represented there in the map. One other metric that is commonly used associated with binary predictions is what you see in the panel here on the bottom right, where this is called the receiver operator curve. And what it's doing is taking from 0 to 100, or 0 to 1, if you will. And at any of those steps from 0 to 1, you can classify. Of course, we classified at 50, but what 50% or 0.5, but we can classify at 1 or 0.1, 0.2, 0.3, and so on. So the idea is that we can see the curve in red is the specific curve for this model under all those classifiers, under all those thresholds for the classification. And then what you can do is compute the area under the curve, which is the AUC value of 0.65, to see how well your model performs under all those classifying conditions. So really, really good model would approximate 1. A model that's not better than random would approximate 0.5. So in this case, we're not doing fantastic, but actually I will tell you that given that what we are predicting here is behavior, we're not even predicting distribution, but behavior, this is actually really good. We have other work we're doing predicting distribution, and we get AUC values of about 0.6, 0.7 as well. So I'm actually quite pleased with this result. One of the more useful uses of the binary classification of the likelihood prediction is in terms of assessing not just the areas where we know that are known as hotspots, but also areas like this one here, where the model is giving us false positives. That is, where the model is saying that there is ARS behavior, but in reality there isn't. And it's really interesting because that particular area between Point Conception and Monterey Bay essentially has excellent oceanographic conditions and seafloor conditions. It really combines really the best sort of metrics in terms of seafloor slope and so on, and temperature. But that area is not drained by any rivers. So coastal upwelling systems are also fed. One important driver of productivity in coastal upwelling systems is the availability of iron that allows phytoplankton to bloom. So a lot of that iron comes from terrestrial sources. This particular area does not have input and is known as what's called HMLC, so high nutrient, low chlorophyll conditions. Those are mostly known from very far offshore areas, but there are some coastal areas where we know HMLC conditions occur, as is the case here. So this is one area where, of course, we would not be able to have predicted that without knowledge of, say, iron or so on. But I find this a useful way of understanding where the model may not be doing as well and why. So moving on to conclusions, the behavioral states that were obtained from one location per day actually capture what we think are the large-scale processes of blue whale movement in the California current ecosystem during the feeding season. And the spatial and the temporal resolution of the predictable variables that were obtained from remote sensing also appeared to capturing some of those processes. So we think that in spite of all the data aggregation and binning and so on, to be able to arrive at this kind of spatial predictions, we are capturing some of those processes there. Although we know that that seasonal binning is also, of course, reducing or smoothing or smearing some of the signal and essentially taking away some degrees of freedom as well. But for a sort of a large-scale approach, I think the framework does pretty well. So ARS behavior does also, just like distribution and so on, are associated with areas of enhanced productivity or favorable feeding conditions. ARS appears to be linked to that, and that is essentially the first demonstration we have that those behavioral states or behavioral modes identified by state-space models do have some underlying ecological basis. So that predicted ARS likelihood was most intense and extensive on the shelf over westward facing slopes, suggesting that that particular ARS behavior is associated with krill aggregation along the slopes, and also with areas of upwelling both on the coast and offshore. And these responses indicate essentially that blue whales are optimizing foraging behavior along these environmental gradients that we examined. So in conclusion, the take-home then is that behavioral state can be used, a useful measure of ecological performance for blue whales during the summer season, or the feeding season. The amazing work that went into making this happen, numerous field crews, research, administrative staff at the MMI, thanks to the availability of satellite data, we have been able to do this. So NASA, NOAA, Coast Watch, and Aviso. The National Marine Fisheries Service Permitting Office has allowed us to conduct this work over the years. Then as far as funding, we have received funding from the Tagging Pacific Pelagics Project, private donors to the MMI, and this particular NASA grant, as well as the Navy Pacific Fleet and the NAVFAC Pacific. Thank you very much. Maybe we can get some lights, or should we? All right, so 1, 2, that was 3, 4, and 5.
So just a kind of a point of clarification. When you were talking about patches and locations and distances in between, what is the difference? You never addressed the difference between a location and a patch. I know a patch was some area, but what were the locations? What did that represent? Because I know that a lot of this is from satellite data.
Right.
So were those physical locations that people sampled from, like went out and observed behavior, or was it from the satellite?
It's all derived from satellite. There's no direct observation. So just to clarify that then, to repeat the question for those who are online was, what is the difference between patches and distance between locations and the terminology? So the what we call ARS patches are consecutive locations that have been identified as being under ARS behavior. So that could be 1, 2, 3, 20 or more locations that are one after the other are consistently classified as ARS. So from the beginning of a track segment that has ARS to the end of that segment, we call that a patch. So that is to correspond with the schematic we had at the beginning, where we had that sort of, buzzing behavior in one area. That would be the patch. Then it just moves linearly until it finds another patch. So the distance between locations, I think I said it was about 40 kilometers on average. I don't remember. I think it was about 40 kilometers. But of course that had a huge range actually. I think it was like from 0.2 to like 200 kilometers between two locations. Does that clarify what you were asking?
Would it be fair to say that transiting is linear behavior and ARS behavior is clustered with a lot of changes in direction? That's what characterizes the cluster behavior. Is that helpful?
Exactly correct.
Yeah, that's definitely helpful. Thank you very much.
All right. Okay, then was Logan?
Yeah, I have a quick question. In terms of sort of how you go about choosing an animal to tag, and if there's biases associated with that, I know it's something we deal with in the Antarctic a lot in terms of developing a non-biased sampling program. We just sort of sample animals opportunistically. And I'm curious as to whether or not those ARS behaviors could be specific depending on the current life history stage of the animal that was tagged, whether it, well, I assume you don't tag juveniles, but if it was a juvenile or a lactating female, or an adult male or a rescue female or something like that, and how those ARS input parameters may change, or if you've been able to even tease any of that information out or look at that at all.
Yeah, no, so the question was whether differences in age or sex or other sort of life history conditions of the individuals might affect how they perform the ARS behaviors. That is likely the case, if anything, because animals that are, say, young animals that are weaned must learn how to forage, and that is a process of experience. So certainly there must be some component there. Similarly, males and females, from separate work we're doing with other tagging devices, we are seeing that males and females tend to do different things, at least in Southern California. So going back to your question for this particular data set, we do not have that information. We do only tag what we considered adult animals. And we only have sex information. Out of that 104, we only have sex information for 24. And they happen to be 12 males and 12 females. So at least the sample is balanced as far as the ones that we know the sex of. And they do occur, obviously, in the same area. We're not just, there doesn't seem to be any indication that there's only males or there's only females in any given area. But that's certainly, it's sort of the next step is what can be done. Right now, I think we're sort of taking a real first cut at what we can possibly extract. It's really, if anything, was pretty involved exercise in extracting signal from a lot of noise. And there's so many ways that you could go about, and likely over time we're going to be mining these data sets in other ways, maybe just taking a subsample of just the males or just the females or certain things and looking at those things. But the thing is that once you start asking that kind of question, your data set really reduces dramatically, both in space, time, and every other condition. So you then, if you're trying to answer questions that are these kind of large scale, you would not have the ability to do that. Who's number two, Marta or Bob?
So I'm wondering if there's any opportunity to introduce bias in your estimates of the hotspots based on the spatial arrangement of your sampling. They were the same 3 locations where you tagged were those hotspots. So, or is it? I'll leave it at that. Is it a chicken and egg or egg thing? Did you sample where you knew the hotspots were,
Right.
or did the sampling in those locations create any bias in determining them as hotspots?
Well, to cut to the chase, the bottom line is that in order to be able to successfully tag a good number of animals during any tagging season, we need to go to areas where they are reliably and found and in good numbers for us. So that means that these operations are very expensive. So in order to be able to do that, we have to select those areas, and oftentimes we have support from aerial spotters that do some surveying just before we go out to sort of get the layout of where the animals are and point at that. So given that, yes, there are issues given the fact that tagging has occurred in areas where they happen to be more abundant, and there is some of that. So we have taken a number of steps to remedy that, and I think what we see here is really truly the underlying pattern. It may be slightly biased, but I think we have done everything we can to do that on a number of fronts. For instance, for this particular subset of data that I analyzed, I took tracks that were longer than 14 days and that covered at least 888 kilometers. So what that is doing is allowing, if a whale wanted to move out of the tagging area, because for the first study we did, we were able to determine what were the average distance between foraging patches and how long it took them to get between them. We excluded any track that was shorter than that to essentially capture if the animal would move throughout the area. So that is essentially actually biasing the sample towards animals that move, that take large movements. Another way that we did that is that in the case of the home range study that LAD did, that particular study actually thrived on the overlap of many animals right on top of each other. In this case, we actually weighted all animals the same so that any given grid cell wouldn't have more weight from locations from just one animal, but all the animals would contribute equally. So just a number of considerations like that, little tweaks here and there to try to minimize that, knowing that tagging data by nature are biased. It is the nature of tagging data that we have to deploy our tags at a particular time and space, and then from there so that the animals have the opportunity to disperse or not.
Isn't it fair to say too that when you tag animals in Southern California, they very frequently wound up at the Farallones and some of those other hotspot places. I mean, they didn't just stay.
Right. That is an excellent point, and I might show something that I hope will address a little bit of that. So if you look at the panel, at the map here in the middle, the color represents the number of tracking days. So since an animal was tagged, how long they have been at liberty for the entire data set. So you see that the areas that have the longest tracks are not at the hotspots. They are somewhere else. And where they are, it took me a little bit to figure it out, but I think I got it, is what Bruce was just saying. So the animals are tagged, so let's just say in the Santa Barbara Channel between, let's say, San Miguel Island and Cape, Point Conception. And over the course of the season, which is maybe 90 days, 100 days, 150 days, whatever, they tend to move northward and then migrate southward. So what you see is that the lighter color up north of Cape Mendocino and toward Oregon, that's sort of toward the end of how far those animals have moved from tagging. At the very bottom, you see another sort of warm or yellow area of long tracking durations. What's going on there is actually animals migrate at the end of the feeding season. They go south to, as you saw, the full range, to all the way down to the tropics, to the equator, and come back. So then what we're seeing is the signature of a few animals that are entering back the California current of the U.S. And then they have very long tracking durations there. So again, it's just a way to demonstrate that we have done what we can with this data to minimize that type of bias. Who's number 2? Martha.
So of your 4 and 5 variables that bubbled to the top, 3 of them are temporally static. The sea surface temperature does change. I was wondering if you look to see if the variability in the sea surface temperature might be driving some of these faults, negatives and positives, and any other thoughts about the temporal variability that you're getting.
The question was whether some of the non-static, that is the seafloor associated variables, so the non-static oceanographic variables that are known to change over time.
No, the non-static.
Yeah, so the oceanographic ones, their variability might have some effect. And I would ask you back, I'm not sure I'm getting what you're trying to say. What kind of variability are you talking about?
In terms of the variability of the values within one grid cell, and whether or not that grid cell, because there's higher variability in sea surface temperature in certain areas, may be contributing to misidentifying what the behavior is in a particular grid cell.
Okay. Right. So that is an interesting question, because I think the final answer, the bottom line answer to this question is that the way you might envision this type of results that I just presented is we're looking at the long-term average summertime long-term average conditions. So in oceanographic or meteorological disciplines, we call that climatologies. So it's sort of what you expect to see in long-term averages, knowing that the average never really happens. Things that generally tend to be above or below. So that would apply generally to every grid cell that we have. Now, I think the question that you brought up about whether some of those grid cells may have more or less variability, let's say in temperature, and how that might affect this, I have not looked at that explicitly, but I'll tell you that by nature, because the hotspots are clearly associated with the most productive areas of the California current. Those areas are driven by upwelling, by the very nature of the pulsed process of upwelling. Those are also the areas of highest variability. In any variable that you look at, temperature, chlorophyll, kinetic energy, whatever, they all have the highest variability. It's inherent in the driving force. So I can tell you right away that that would certainly be the case. We would see higher variance in those grid cells just because of that. But I would love to hear more thoughts about that. If we can chat later about that, it'd be great. Who's next?
Getting back to the ship strikes.
Right.
And I'm curious about the seasonality of the hotspots in relation to avoiding ship strikes. In other words, similar to what happened, with the North Atlantic, would it be something that this is a problem in the summer but not in the winter, and that changing ship traffic during that season might have some positive impacts, or not have negative impacts?
Yes. So the question is related to the fact that some of the blue whale hotspots happen to occur in areas of heavy marine traffic, and there are a number of known mortalities that are being caused by ships, and they are a cause of concern for the recovery of the species. And whether some knowledge about the temporal seasonal evolution of these hotspots, can we say something about how to mitigate that situation? So because of the nature of the data we have, in terms of, as much as we're always striking a balance between aggregating the data enough to be able to capture some type of spatial pattern that is representative, but also maintaining some level of resolution. The work that Lad Irvine did here with the home range overlap in relation to shipping lanes actually had a temporal component within the foraging season. So the blue whales are very seasonal off the west coast of the U.S. They show up from south to move to north, then move back south and leave. Sometimes they leave sort of an offshore route, but essentially that's the general north to south pattern. It's like move up north and move up south. So certainly the time they are hitting Southern California is earlier than the time they're hitting Central California, and so on. So in terms of management applications, where you could think about it in terms of a time area closure or some kind of regime like that, that information certainly is valid. And as part of that paper, we gave specific recommendations based on the data we had about the timing of this, what would make the most sense from the whale's perspective. Of course, there's all kinds of considerations from the stakeholders' perspective, economics, shipping agencies, fuel conservation. But as you mentioned, from the East Coast, where they also have implemented shifting in the shipping lanes and the timing and the speeds and so on, there are stakeholder processes going on right now of the West Coast of the United States, where this information is being incorporated into how best to maybe improve the flows of these shipping lanes, which is what, by the way, which is what you see in the black lines, is for instance, the San Francisco area port has 3-axis lanes where all the ships just funnel through those 3 areas. The Santa Barbara Channel has just 1. Yeah, depending, for instance, the northernmost shipping lane here in the San Francisco Bay Area goes right through the area of largest aggregations, and that could be a good basis for maybe altering that. So I would refer you to the paper where we, as best we could, based on this data, we generated some recommendations. They are being considered by the people who are in this process, and of course they have to take other considerations into account, but that's the best answer we have.
Daniel, could I chime in one thing here?
Of course, yes.
Behavioral on people. And the Southern California one, the county of Santa Barbara was willing to pay $1,200 per ship if they would slow down from 30 to 35 knots down to 15 knots. And that was based on human health, because you got more particulates from the exhaust when you were going at high speed than you were going at low speed. And they thought, for human health reasons, they thought the 30 to 35 ships a day going by, that would be worth an investment for them. Despite offering that money, none of the shipping companies took it. But when this paper came out, it only took 6 weeks for 6 of the 9 major shipping companies to drop their speed.
Really? That's great.
Now, they took the money too. But I guess the point I want to make is there's some iconic species that can drive things on a PR basis as much as anything else. And when we were down there doing work for a month and a half geographic, we encountered 5 dead whales in 1 month. I think that was the highest that's ever been known, but the animals aren't there during the winter. So, but ironically, one of the alternate routes is south of the northern islands. It's a more direct route into Los Angeles, saves fuel, gets it away from human populations for the pollution issues, and would save whales. But it's also a military area of military exercises. So it's one of those stakeholders has a higher trump card than some of the others.
Yeah. The military, of course.
Thank you. Great. So, Scarlett.
So there's a couple questions from the online group. Was there a particular reason blue whales were chosen for this study, or has this technology been used for other species?
Was this particular technology only used for blue whales or used in other species? We have certainly used it in something like 8 or 10 different whale species in very different populations. That essentially has been Bruce's career here at Oregon State University. So I think it would be fair to say that our largest data sets come from blue whales, sperm whales, and humpback whales. But that said, Bruce has tracked right whales in the North Atlantic and the South Atlantic, fin whales in the Mediterranean, blue whales in southern Chile, and
Bowheads in the Arctic.
Greg here did his master's with bowhead whales in the Arctic. Of course, another great data set we have is gray whales, including eastern gray whales, western gray whales, and Pacific Coast Feeding Group gray whales. Am I forgetting anything, Bruce?
And the second one was, is there any assessment of potential impacts from El Nino events to the movements and distribution of the animals during this long sampling period?
Great question. So question was whether and if events like El Nino would have an impact on this study. And absolutely, certainly for sure. We see very strong differences in years, like this most recent last 2 years when we had a strong El Nino and a warm blob and good conditions and so on. We see very differences in the aggregations of the whales themselves, where they are, how many there are, also in their body condition, their behavior. So all that is there. And clearly for a whale that is tied, essentially a species that is tied so close to the base of the food chain, the food chain that blue whales exploit is very, very short. It's one of the sort of the best case studies is you have coastal upwelling, diatom blooms, krill, whales. So we know that all along that track, with that chain, any event like El Nino, Pacific Decadal Oscillation, and so on is going to have an impact of that. So as I was mentioning earlier to the question about the variability in environmental conditions in a grid cell, the approach that we took for this particular study was to look at the long-term averages rather than the variability in time. We certainly could try and take a year that was considered La Nina and a year that was considered El Nino and see how they differ. The problem is that by slicing it by an individual year, immediately we lose a lot of resolution in the data. We just don't have any spatial resolution. So being able to say something about that would be difficult. So the to answer the question then, again, one is we're looking at what we might expect as an average that implicitly incorporates good conditions, bad conditions, El Nino, La Nina, and so on. But further than that, we also know that in addition to these interannual events, we have the interdecadal events, and decadal events like the Pacific Decadal Oscillation and so on. And those conditions, we know that they changed in 1998, and they were persistently good, actually, good throughout the 2000s until about 2011 or 12 when they shifted. That was because the Pacific Decadal Oscillation went into a negative phase. So what gave me confidence in taking this particular set of years for this analysis is that at least at the very low frequency variability of the Pacific Decadal Oscillation, we're talking about a period of similar conditions across that, knowing that there were, of course, some years with El Nino or La Nina and so on that are overimposed in that. That was the first question, right? Okay. Do you have another one?
No, that was the second.
Oh, the second one. Yeah. Okay.
Taking that a little bit further down the line, do you think that there's any capacity for this model to predict changes in behavior and climate change, changing oceanic conditions? If there's such kind of upwelling to drive food web, we see dry fluctuations in the upwelling patterns, will that make research?
Yeah. The question was if we can extend this type of predictions to climate change scenarios. And that certainly is a really good application of this. I would say yes and no, or yes with caution. The main reason about the caution is that oftentimes we are able to derive relationships that hold over, even over a decade like in this study, so we can more or less say that we have at least incorporated a long enough time period to sort of sample a variety of conditions, but the predictions that we get, they're all constrained or restricted to that type of association. Sometimes when you talk about shifts like climate change, where everything gets really shifted, you don't have, it doesn't mean that those relationships will stay the same. They might break away. So we could be generating predictions using future scenarios with the underlying assumption that the associations remain. There are people who specialize on that, and I think there's, thoughts, opinions in favor and against doing something like that. So I think as an exercise, of course, it's a good thing to try and come up with what could happen. We do certainly, as you said, we might expect that the areas where they go, because upwelling areas might get strongly disturbed. First of all, these hotspots, which at this point are essentially static in space, they may be obliterated. And we kind of see that in a way in the last El Nino, where the southernmost hotspot here in the Santa Barbara Channel, they were not there in 2015. So we know that can happen. But as far as being able to use those relationships to predict about climate scenarios, I would be a little careful with that. I don't think we're just there yet to be able to have a real confidence in that. But as an exercise, sure, why not? All right. Oh,
A follow-up question to that then. So what would it take to get us to that?
So, well, I think it would take, it would essentially take tracking over longer periods, and where we are in a good position to do that is that in the last 3 years, the Navy has funded us to do additional tagging of blue whales. So in fact, in the last 3 years, we're already up at 64, 65 blue whales that we have tagged in the last 3 years. This is new tracks with newer technology, better tracks, better technology. And as I mentioned earlier, the changes between Pacific Decadal Oscillation sign or phase can probably be used as analogous to climate change type scenarios, maybe not as dramatic, of course, but essentially we could take our new data from the newer data set, the newer years, and run separate models under the new Pacific Decadal Oscillation phase, which is a warm phase that started out in 2011, 2012, and see if we're capturing similar or different patterns. So that is the first step that we could actually do with the data we have. Beyond that, I think it's about having better climate change scenarios predictions, which are always the, you have to take them with a grain of salt, and they're usually really good for sort of large-scale problems of global impacts on things rather than local things like these hotspots that are at the larger scale of the basin or global scale, they're pretty small. So I would trust that as well.
So do you match that up with in the plankton samples? Or?
Well, that is what's really challenging, and it's only getting worse and worse. Plankton sampling is extremely expensive, demanding. There isn't any plankton collection at the scale of this ecosystem that we could use on a matched space-time basis to be able to predict how prey may be changing in relation to well behavior, for example. So that is extremely challenging.
But you could in those hotspots.
Even at the scale of the hotspots, I mean, unless a university is able to, or a government agency like NOAA is able to decide that they're going to go and sample these hotspots intensively over the course of the season, year after year after year, that is hard. So there are actually Cal Coffee program, there's the NOAA Rockfish program out of Santa Cruz that specifically sample krill. And guess what? Their surveys do not coincide at all. There's actually some good krill data for a stretch of the coast between Santa Cruz or maybe Central California to almost Point Conception. They don't, at this point, they don't match us in time with the blue whale presence there at all. So, and those are sort of some of the best data sets we have in existence. The Cal Coffee program, likewise, for a restricted area, also has some pretty good, and probably the best sampling, if anything. But likewise, I think in that case, some of the issues have to do with the fact that the krill samples have not been analyzed at the scale that we would want, because they actually have analyzed sort of the long-term trend in krill, for instance, but it's integrated over the entire Southern California body. So it's not resolved in space, it's just one integration in space rather than what we have here, which is much more resolved in space.
Danielle, could I add something?
Yeah.
Because I think he's in a difficult position because he's showing you a group of data that's been collected some time ago, ended in 2008. And what you were asking several of you about El Niños and the change in decadal oscillation are recent issues. And he mentioned in the last 3 years we've been tagging whales for the Navy. We've really done very, very well in a time when we would normally have avoided tagging because the animals weren't doing well. I mean, this year when the tag team went out, about 80% of the blue whales they approached in Southern California were emaciated. 2 years of warm water blob and El Nino, I mean, these populations really been hit hard. They eat krill. They don't do anything else. Yeah. And so they've really been hit. And I think I'll go a little farther than he might, just because it's probably my liberty to do that.
Go for it.
Say that I think we've really stumbled on something very, very exciting in terms of for blue whales as an indicator species of what's going on at that lower, that productivity level. And I think it is probably a good indicator of a good parallel to warming. I mean, obviously there needs to be more samples and all that kind of stuff. We've never had the luxury of having this kind of data. And interestingly, when the second year we were going out, the Navy said, oh, we've only got money for 7 tags. And we went, oh my God, this is the year where you really ought to do a lot. And so we pushed them and said, hey, we've got permits, we've got time, we've got ship times. What if we've got tags? Let us give you the tags and you pay us back when the new fiscal year comes. And we told them why it was so important and they bought into it. And that year I think we tagged 35 whales or something like that. So, I mean, some of this is opportunistic, but I think we've got a tiger by the tail here in terms of very, very strong signature. And it's pretty exciting. And I might add that whales didn't go as far north.
Yeah.
And when these animals come up from the south and they're hungry, they stop at where there's food. If there's food, they're going to stay there really well. And if food fails, they go farther north typically. Over here we had several years where it failed. We think they probably weren't in condition to do that, to take the risk. They probably had to make do with foraging in areas where there was low productivity, but it was assured. They could trade calories and live. Whereas if they went far, they took risks, and if there weren't things there that could really compromise their existence. It was an interesting strategy trade-off. I mean, I think we've still got something that's exciting ecologically. If I overextended, you can tell me.
That's why he's so good at what he does.
Daniel, one more real quick.
Yeah, Jim.
Do you have any other data set, like pinwheels, for example, that could either be a parallel comparative study or a complementary study to this?
The question was if we have complementary data from maybe other species in the same ecosystem, for instance, fin whales. I did not plant the question, but that is the most exciting question that at least for me. Because yes, Bruce historically has tagged a fin whale here, a fin whale there in this particular area. Prior to the last 3 years when we received Navy funding specifically for fin whales. Fin whales are ecologically apparently similar to blue whales. They are morphologically similar. They're just a little smaller. They're very difficult, very challenging to tag. They're super fast and erratic and a challenge. But thanks to the amazing field crew that we have, they have tagged something in the order of maybe 35, 40 fin whales in the last 3 years. So now we have a really nice data set, and the preliminary analysis that we have done comparing fin and blue whales tagged in the same area at the same time show very, very interesting differences between the two, where they go, at time where they go. So stay tuned. Maybe in a year we'll give another seminar comparing the two. It's really exciting because almost nothing is known about fin whales, really.
They are switch hitters too.
Yeah.
I mean, they can feed on more than krill.
Very good point.
But I'll share one thing with you. The first animal we tagged long duration, 380 days or something close to a year. The animal did the opposite of what you'd expect. Instead of going south for the winter, it wintered in the Gulf of California, Gulf of Alaska. Yeah, and it did a migration in the spring. South of the tip of Baja, but 1,000 kilometers offshore, and it never slowed down. Never had any ARS behavior for either feeding or reproduction, and then went back up to the Gulf of Alaska. So are we excited about that? Fin whales have got some, they haven't read the book yet.
Are you sure it's a whale?
Yeah.
All right, can we go ahead and thank you?
Thank you, everybody.
Can I ask a question? On your confusion table? Yeah. Those were raw data points that you were comparing?
Yeah, I was going to
model
I was going to ask the
results,
ship
or-
strike, and then that guy asked it.
They
And then I was
were
going to ask about
observation
tagging, and then this
Okay,
guy
and
asked
were
it. It
the-
was like,
Oh,
okay,
confusing
well,
because
all
you're trying
right,
to
but yeah, I was going to ask him.
Compare how the model
Yeah.
is doing
Oh, by the way,
relative to the observation.
I
And were
wrote
those because
something
you're low
because you weren't
end,
here
were
last
those
week, and that's why I did it in
observations
case.
used in the model?
Oh, yeah.
Oh, you're on the.
So what does that do to your statistical?
I heard your question.
It's incorporated in the process, but essentially you have your
Wait,
observations.
wait.
They're what they are.
Who was in charge of it?
You
Oh, okay, that
generate
makes sense.
a likelihood.
Ah.
And then it's essentially
That's why I was like, okay, whatever, it doesn't matter, it's easy.
predictions
But
to the original observations.
So,
Like your predictions are done off your Monte Carlo?
No, Monte Carlo. This is analogous to a logistic regression.
Okay.
So this is built into the model. Like a logistic regression is basically the same confusion between how you compare your observed presences versus what the model did. So of course the predictions are based on the observations, but it's essentially saying that if you have such observations, the perfect world you'd have the same prediction. Like if you have presence, you would have presence. But you don't do that. In the prediction, you have some precedents. So some positives, true positives and false positives. Why? Because models are never perfect.
Right. And if you had
It's
a
a
bigger,
simplification of the real world. So you expect that by nature you will have some failure in the model. So it's a way of assessing failure in the model.
If you had a larger N, would you pull out a data set from that original data
No,
set?
I know what you're saying. You're asking about training and validation.
Yeah, doing more of a validation
Right.
type
So
of thing.
I thought about doing that. Unfortunately, at this time, something that I can do now is with the new data. But at this time, just based on this 1999 to 2000, 10 years of data, this is the best we can do. So I could not afford to even
Take out the 10%.
Right, no, and also because the purpose of this models always have to have a specific purpose. A lot of people use models for the very specific approach of predicting something in a forecast way, as in saying, I need to know what it's going to do tomorrow because I'm going to use it operationally. In this case, our models are exploratory.
Right, so
they're
it's
just trying to tease out things. But now that we have this, and because we have very good recent data set that did not make it into this approach, because I started this study some years ago, then I can use the new data set as a testing data set, how it performs.
Right, and then you can do that same comparison and see if it predicts that same. Okay.
Right.
And so that adds something to your.
It would just certainly validate, at this point I have this, somebody might say, well, but that model is good for
predicting what you already know.
What you have, and what you have.
Yeah.
But what if there were other times? How would it do? Would it do as well or not? Well, I would say, well, because it's over such a long time, we're integrated, over-integrating in different conditions, but on the other hand, things changed since the last time.
Right, so your new data is going to be outside of those basic parameters, and so it's going to be...
It's going to help. It's going to say, that model performs equally well or performs terrible.
Right, only when you have these set parameters. Okay, awesome. Thank you for that. Can I shut you down out of here?
I'm done here, so
okay.
I can let you take control.
Okay. Thank you for that. Do you want me to keep recording? We're done. Okay.
How you doing?
Hey, Aiden.
Oh. Yeah, dude.